Hello, AP Biology students. It's Mr. Sanger, screencasting from the storeroom. Today we're going to talk about a lab called Factors Affecting Photosynthesis. And the objectives of this lab is you are going to measure the rate of photosynthesis. Again, you're going to review and practice scientific process skills by designing. This time, though, we're up in the ante a little bit, and that you're going to design an experiment by choosing some different variables to manipulate. And then third, you're going to study the effect of various factors on photosynthesis. And that factor is up to you and your group. It could be the effects of light color, light intensity, carbon dioxide concentration, light exposure, or anything else you come up with. So let's go through some background and then we'll talk about what you're going to be doing. So the first thing that's going to happen is your teacher will isolate chloroplasts from spinach leaves. And we're going to use that to actually carry out photosynthesis in a lab setting. And I think it's kind of interesting. We literally are going to take some spinach from the grocery store, stick it in a blender with a little solvent, and we grind it all up and filter it. And it's loaded with chloroplasts. So that nice green color that you see in spinach is because of the chloroplasts. So kind of cool. To measure how fast photosynthesis is happening, we are going to be taking advantage of the light-dependent reactions of photosynthesis. So I want to highlight a key step here. You may recall that in the light-dependent reactions, light excites electrons. Those electrons are passed down these electron transport chain here. And then this electron in photosystem 1 is passed to NADP plus reductase. And there, NADP plus accepts the electron to become NADPH. And this NADPH is later used to actually build a carbohydrate out of CO2. So in this lab, what we're going to do is we're going to replace the NADP plus with a dye called DPIP. And the interesting thing about DPIP is that it changes color based on whether or not it is bound to an electron. So here you see a bluish color for DPIP, but if it accepts an electron and becomes reduced, the color actually becomes lighter. And so what this means is the oxidized DPIP is blue, the reduced DPIP is clear, and so as photosynthesis takes place, this blue DPIP will be accepting electrons and becoming clear. So at this point, we need to ask ourselves, all right, what tool do we have that could measure a color change in a reaction? And hopefully, if you think back on an earlier lab, you might think, hey, wait, we had those fancy colorimeters. Couldn't we use those? And the answer is yes, we can use those. And so that is what we're going to be using to measure this color change. So just like before, I'll remind you when we're using these colorimeters, make sure you only handle the cuvettes by the ridged edges to prevent your fingerprints from getting on the part that the light passes through. You want to use these little Kim wipes to clean the clear side off before using, so you can get rid of any potential smudges. Make sure you have a cap on the cuvette before putting it in your colorimeter, and then invert to mix before each reading. So those are just important things to keep in mind whenever you use these tools. So I want you to think about this a little bit. What should happen to absorbance over time as photosynthesis occurs? So as we use this blue dye and it becomes clear, should absorbance increase, more light be absorbed, or less light be absorbed? Think about it. You got an answer? Hopefully you said, whoops, there's no answer appearing. Hopefully you said that absorbance should go down, because if we go from blue to clear, more light will be able to pass through, and so less light is being absorbed. And so we should see a graph over time where absorbance is actually decreasing as this process takes place. So how are you going to design an experiment around all this? Well, here's the gist of what you're going to be doing. You're going to be setting up several cuvettes with slightly different experimental conditions, and you're going to place them behind a beaker of water, which is between them and a lamp. The lamp is a light source, and the purpose of the beaker of water is to absorb any heat that light is given off, because the heat, as we know, can affect the rates of reaction. If it's intense enough, it could even denature the enzymes involved in photosynthesis, which is not good if we're trying to get photosynthesis to happen. So you're going to set this up. And then the question is, what goes in each of these cuvettes? And that is where you and your group need to make some decisions. So I'm going to have all of you guys do a control cuvette where you add 1 ml of phosphate buffer. That basically creates the proper pH for photosynthesis to occur. 1 ml of water, 1 ml of the DPIP stuff, and then 3 drops of chloroplast. That is your control trial. You are then going to need to make 3 experimental cuvettes where you manipulate. How many variables would be good to manipulate? 1. Hopefully you said 1. 1 of these variables in some way. So what are some examples of what you might manipulate? Well, you could change the light color. We have different colored light bulbs, and so you could have one trial be red light, one trial be blue light, one trial be green light. You could change light intensity by altering the distance of the cuvettes from your light source. So we have meter sticks, and so you could say, all right, we're going to put this cuvette right up against that beaker of water. This one we're going to put a few inches behind that, and this one a few inches behind that, and you could decide how far back you want. You could even put one, well, actually, no, we could also measure light exposure by using screens. We have these little screens, and you could sort of wrap the cuvette in 1 screen and then wrap another one in 2 screens, wrap another one in 3. You could wrap one in aluminum foil to prevent any light from getting through there. You could change the CO2 concentration. So remember, in your control cuvette, you're adding 1 ml of water. And so you could add carbon dioxide in the form of baking soda, which breaks down into carbon dioxide. So instead of adding 1 ml of water, you could add 1 ml of carbon dioxide, or half a ml of carbon dioxide, half a ml of water, 1/4 ml carbon dioxide, 3/4 ml of water, keeping the volume of that 1 ml of water constant. You could also change concentrations of the phosphate buffer, the DPIP, or the chloroplast in some way if you want. Or if there's something that we haven't thought about, you could come up with your own idea. The key is you want to be clear about what you're testing. And so in years past, we always struggle because groups don't always tell us what they are manipulating. So make sure you change all 4 of these column headers. If you're measuring the light intensity, you might say control cuvette, 1 inch from beaker. This one could be 3 inches, 5 inches, 7 inches, whatever it is. White light, blue light, green light, red light. So you want to be specific so that you can add your titles in there. Okay, so you're going to design an experiment and form a hypothesis and make some predictions. And then on lab day, you are going to come in the room and you are going to prepare all 4 of your cuvettes based on what your pre-lab tells you to do. So it could be you're changing the concentration of something here. It could be you're setting up meter sticks and distances and putting little tape down to specify where those cuvettes go. It could be you're getting the different colored lights set up. Whatever it is, you're basically going to get everything all set up, but you are not going to add the chloroplast yet. The chloroplasts are kind of like the enzymes from that last lab we did, where as soon as you add them, reactions are going to start happening. So you're going to wait on the chloroplast, but get everything else set up as quickly and efficiently as possible. So during your pre-lab, you want to consider, how do we get this set up, and maybe even go into the lab area and start to think it through so that you could just come in on lab day and boom, 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 get everything going. So you have as much time as possible for data collection. Then, you're going to use that heat sink that I mentioned, that beaker of water. And then just like before, you're going to do the calibration of the colorimeter, where you just basically put in your control trial and then push that calibrate button. And then you're going to begin collecting data. Now, note that you do not need to set anything up for data collection, and you don't need to press play on the LabQuest. All you're going to do is put the cuvette in at a certain time, read the measurement, which will be displayed, I think, actually in the big box, and then take it out. And then when you're ready for the next reading, you're going to put the next cuvette in and just read it. So there's no hitting play. It's not going to generate a graph. You're just going to use the values and record those as we go. So the way it's going to work, and this is important to understand before lab day so that you can just get it going as soon as possible, because you'll see we're trying to collect 20 minutes of data. So if you take a long time to get everything set up, you're not going to be able to get all of that. Okay, so here's how it's going to work. You're going to have everything set up, and you're going to take your control cuvette. Again, your lab report, it will document what that control is or what it looks like. And then you are going to start a timer at the instant that you add chloroplast to that cuvette. So at time 0, you add chloroplast to that cuvette, and it's going. Exactly 1 minute later, you're going to add chloroplast to this cuvette. Exactly 1 minute after that, you're going to add chloroplast to this. And exactly 1 minute after that, you're going to add chloroplast to this. Okay, so times 0, time 1, time 2, times 3. Minute 4, kick back, relax, do whatever you like. But then minute 5, you are going to be needing to get a reading on the absorbance for your control cuvette. By the way, you need to get that reading at the beginning as well. So add the chloroplast, take your reading, and then go. So minute 0, minute 1, minute 2, minute 3, minute 4, you rest. Minute 5, you take that cuvette, you put it in the colorimeter, and you read the absorbance again. Then minute 6, you read this one. Minute 7, minute 8, minute 9, you rest. Minute 10, read this one. Minute 11, minute 12, minute 13, minute 14, you rest. And the idea is, even though you're kind of off by a minute for each of these trials, for each trial, since you started this trial at minute 1, minute 6 is actually 5 minutes of time. So by doing this, you're able to sort of go along this table minute by minute, but keeping sure that your distance between trials is in fact 5 minutes. So again, make sure you understand this process, make sure your group has a system in place to make this happen, and make sure you're ready to go, because the timing of this is really critical. And once the chloroplasts are added, the reactions go on whether you're ready for it or not. So to get the best possible data, you want to have all your ducks in a row, add the chloroplast with that timer going, take your reading, and go. All right. You are then going to enter your data into a Google Sheet, and you're going to perform what is called a linear regression, which is basically a slope. And so you're going to figure out the slope of your lines, and then you're going to record those slopes as a rate of photosynthesis. And since absorbance is going down, you will have, I'm trying the mirroring thing, is that the right direction? I don't know, it's going down. Since it's going down, your slopes will be negative. And so you're going to record this as an absolute value to avoid that confusion of the negative. So the steeper the slope, the higher the absolute value number will be. And you can kind of calculate how the rate of photosynthesis varied between your 3 trials. So you're going to collect the data, you're going to graph it, you're going to get the slope of that, and you're basically going to record that slope. Okay, so pretty complicated lab, but I think you guys are ready. I believe in you. And just a reminder of these science practices in AP Biology, and you can see we are actively engaged in all of these consistently throughout the course. So that's all I've got. We'll see you on lab day. Good luck to you.